What are antibodies in Parkinson's immunotherapy? Have you ever wondered how our immune system can be harnessed to fight diseases like Parkinson's? Antibodies play a crucial role in this exciting area of research, known as immunotherapy. In the context of Parkinson's disease, antibodies are specialized proteins designed to recognize and bind to abnormal forms of a protein called alpha-synuclein. This protein is central to the pathology of Parkinson's disease, as it tends to accumulate and form toxic clumps in the brain, disrupting normal neuronal function. Immunotherapy aims to reduce or prevent this harmful aggregation. There are two main strategies for using antibodies in this context. The first is called active immunization. In this approach, patients are vaccinated with alpha-synuclein or its fragments. This vaccination stimulates the immune system to produce its own antibodies against the abnormal forms of alpha-synuclein. Animal studies have shown that this method can reduce the deposits of alpha-synuclein and even improve behavioral deficits, suggesting it might slow down the progression of the disease. The second strategy is known as passive immunization. This involves directly administering laboratory-produced monoclonal antibodies that target alpha-synuclein. These antibodies can bind to the extracellular forms of alpha-synuclein, which is thought to spread pathology between neurons in a way similar to prions. By binding to these proteins, the antibodies help promote their clearance from the brain, likely through the activation of microglial cells. Microglia are the brain's immune cells, and they play a vital role in engulfing and degrading harmful substances. The role of microglia is particularly important in Parkinson's immunotherapy. When antibodies bind to alpha-synuclein aggregates, they facilitate the recognition and engulfing of these aggregates by microglia. This process can help reduce the accumulation of toxic proteins and potentially limit neuroinflammation and neuronal damage. Currently, several antibodies targeting different regions of alpha-synuclein are being tested in clinical trials. These treatments have generally shown safety and the ability to induce an immune response. However, larger trials are still needed to demonstrate significant slowing of disease progression. Research continues to focus on optimizing the design of these antibodies, improving delivery methods to ensure they can cross the blood-brain barrier, and selecting the right patients for treatment. In summary, antibodies in Parkinson's immunotherapy serve as targeted agents against the pathological forms of alpha-synuclein. They either stimulate the immune system to produce these antibodies or are administered directly to promote the clearance of toxic protein aggregates. This approach holds promise but remains under active investigation. Patients interested in this emerging therapy should consult healthcare professionals for personalized advice and consider it as an experimental approach rather than a standard treatment at this time.